All right, so I'm going to then begin the symposium by giving the first talk, since I'm already up here. You see my title, Kissing Cousins, Evolution of Host Choice in Eastern Mistletoe. So the first question we're going to have to ask is, what is a mistletoe? And there's my definition at the top, and if you read Geach, well, I guess it's a mistletoe. You mean he was a good kisser? No, he was a parasitic fungus. The born loser, you know, Webster lover, well, I believe Webster says it's a parasitic plant that grows on trees. Leave it to old Noah to kill the mood. Well, actually, the born loser here is closer to the real definition of what a mistletoe is. It's not a fungus. It is a flowering plant, and it is parasitic. And so we know about mistletoe, many of us, from the tradition of kissing under the mistletoe at the Yuletide, Christmas time, and midwinter. This idea probably stems from 16th century England. It goes back quite a ways, and it even dates back further than that in terms of the Druids, who actually had some pagan ceremonies associated with mistletoe, human sacrifice, and some other nasty things. But I want to point this particular, these two little cartoons out because there are a lot of misconceptions about mistletoe. And let's look over here on this one. By the way, for the speakers, if you use the pointer, that will be recorded on your PowerPoint. If you use this, it won't. Okay. Here we see mistletoe hanging up there. That's correct. It has white berries, opposite simple margined leaves, entire margined leaves. And then down here, oh, what do we have? We have white berries on a spiny margined leaf. Over here we have red berries on a simple or entire margined leaf. These are hybrids, okay? Mistletoe looks like this. Holly looks like this, right? So people are constantly mixing up mistletoe and holly, okay? So that you guys won't do that, I brought in some mistletoe, so you can pass that around. Don't eat it. If you've never seen mistletoe, here's your chance. Isn't that pretty? Okay. The berries are falling all over the place here. If you feel so inclined, take a berry between your fingers and crush it, and you'll get a nice sticky viscid mistletoe seed, and we'll be talking about those very soon in terms of the interesting biology that goes on with this plant. But first, let's talk about parasitic plants. This is a picture of sort of the general flavors of parasites. We have what are called hemi and holo parasites. The hemis, as you see from the shading, are green, like this mistletoe. They photosynthesize. The holos, over here, are yellow. They don't have any chlorophyll. They don't photosynthesize. All of our mistletoes are in the hemiparasitic group, but not all mistletoes derive from the same lineage. They've actually evolved 5 different times in flowering plants. And those 5 times are shown by the family names that we see right up there. The biggest family of mistletoes is this one called Loranthaceae, about 75 genera, 1,000 species. Big, beautiful tropical plants with gorgeous red bird-pollinated flowers. Unfortunately, that's not what you're holding in your hand there. You're holding a very tiny flowered member of the Viscaceae. 2 different families, Loranthaceae and Viscaceae. So for us, for Southern Illinois mistletoe, we're in the Viscaceae. That's an interesting, it moved the in the case of way over there. But this is a quote from Darwin. Darwin actually made some comments about mistletoe. In fact, the mistletoe that he was looking at in England was the one in the background there. That's a Viscum album, which looks very much like the one that's being passed around. It's the one that the Druids were interested in too. And as you can see, Darwin's quote is a very long sentence, the first one there, but it ends with, "It is therefore of the highest importance to gain a clear insight into the means of modification and co-adaptation." Basically, he's saying that this organism is intimately tied with many other organisms, from the insects that pollinate it to the host plants that it's growing on. And in fact, this was pretty much my call to arms for working on mistletoes, which I've been doing for, gosh, nearly 30 years now, to follow this sort of an idea, to look at the relationships of mistletoes among themselves and their host plants. So let's look at what we have right here in Southern Illinois. This is a picture of a nicely infested or terribly infested, whichever your perspective is, silver maple tree growing just north of 147 on Route 3, just ways from Murfreesboro. You can see that the mistletoe plants are All of these green clumps, it's wintertime, so the silver maple doesn't have any leaves. So lots and lots of mistletoe all over these silver maples. It, by the way, does also grow south of here much more abundantly. If you go down to Metropolis, Paducah, Cairo, you'll see it all over the city trees. Lots and lots of mistletoe. We're right at the northern boundary here of this species. Although I tell my students and friends global warming might actually help this. We can see mistletoe marching further north. That would be great for me. So this is what the plant looks like. This is Phorandendron leucarpum. Here's the plant you're actually looking at. It's a piece off of this one. You can see here's the host branch and this very large clump, about this big around, growing there. The plants are dioecious, which means they have separate male and female plants. So this is what the tiny little male flowers look like. The one you're holding is obviously a female because it's bearing fruits. The fruits are these white berries that have a viscid seed on the inside. The family name Viscaceae comes from the fact that these are viscid or very, very sticky. Now the hosts of this plant are varied. We have a partial list here of the number of trees that Phorandendron leucarpum grows on. That's 50 or so names out of about 150 that I have in my database that it occurs on. So you would probably say, given this is a very host generalist or a non-specific plant. But stay tuned, things are going to be a little more complicated and interesting than that. This map shows the host preference by state. So here in Illinois, even though the one you have came off silver maple, the most common host is American elm. And that's true in Missouri, Arkansas, and so on. But as you go to the East Coast, you'll see that in Virginia and North Carolina, it likes Acer rubrum. In the South, it loves Quercus nigra, the water oak. So the preferences, if you will, host preferences, change as this parasite, as we look across the range of this parasite in the eastern U.S. Taxonomically, Phorandendron leucarpum is actually a complex, which means that it has a lot of different taxa that are closely related to it, possibly the same species. And the most recent monograph in 2003 by Job Kite actually named 4 subspecies of Phorandendron leucarpum. And you can see the shading of where these occur. So we have subspecies leucarpum here in southern Illinois, the East Coast all the same. But as we get out west, we get into what's called subspecies macrophyllum. It was named as a species. But look at Texas, what Kite called the taxonomist nightmare. It's where 3 of the 4 subspecies all co-occur and even occur with each other in this central part of Texas. I'm going to be showing you some data from my colleague down in Huntsville. They live very, very close to this interesting overlap zone for the subspecies. Now, what would happen if we looked at these subspecies with molecular data? And this is part of a tree that was collected by a friend of mine and colleague, Vanessa Ashworth. She combined ribosomal DNA sequences to generate this tree, and we can see that we have 3 of the 4 subspecies listed here. Serotinum is really the leucarpum today. It's changed its name, tomentosum, and so on. But the point of this tree is that they're very closely related. Using this relatively conservative gene, the ribosomal gene, we don't have very many branch length differences between these. So that was the first study in the 2000 or so. And then came Chris Randall and his students down in Huntsville, Texas, at Sam Houston State University. Chris has been really pioneering a lot of interesting studies on mistletoes. He gets his students to climb up in the trees. It's all over their campus, growing on mainly water oak and several others. So he's done demographic work, molecular work, even some interesting seed biology that I'll show you in just a second. But one of his master's students, Angela Hawkins, shown here holding some mistletoe, did some very important work to help address this issue of host selection in mistletoe and basically genetic differentiation. So let's start with the morphology. She did what's called a principal components analysis, PCA, where you basically measure leaves, pubescence, a variety of morphological characters, and you analyze them to determine the variance in the data set and also the presence of groupings. And I don't know whether you can tell that or not, but it's a little bit difficult to discern any major groups based upon the colors of the symbols here for our 4 subspecies. For example, look at the green triangle of subspecies leucarpum. It encompasses pretty much the range of the whole cluster in its entirety. So basically, Job Kite's monograph that's recognizing these 4 subspecies can't be followed very well. You can't really discern these morphologically very well. And that's, again, from morphology. But Vanessa went on, and not Vanessa, Angela went on and actually looked at microsatellites, another form of analysis using very rapidly evolving DNA. And what she has here is the results from looking at 96 individuals from 10 populations, collected from New Jersey all the way to California, and looking at basically 4 loci, 4 primer pairs in the microsats. This top bar represents the color that one would expect if it was morphology, and here's the code to the 4 subspecies. And this bottom rectangle that has 4 different colors in it is the assignment based upon microsatellites. And as you can see, in some cases, they agree that the majority of this particular bar, it's a percentage of the total of the bar, is yellow, and that is for subspecies Leucarpum, and it indeed morphologically was Leucarpum. But look at these other ones where it conflicts with that. Like right here, we have mostly a green bar, subspecies Angustifolium, but we would have morphologically called it subspecies Leucarpum. So basically, there's varying levels of admixture that's apparent in all these individuals, and they contain alleles from ancestral populations other than their own. So we have a lot of genetic complexity going on here that's determined by these microsats. So again, not very clear in terms of these subspecies. So a little pardon to Strother Martin from the, hopefully the older people in the audience know the movie Cool Hand Luke. My daughter never had heard of it. But basically what we've got here is a failure to differentiate. Did I do that okay? What was the original quote? A failure to communicate. You got it. Okay. Now let's go back to this picture. You maybe hadn't noticed, but this is covered with mistletoe, but what about these trees over here? Right? Nothing. I surveyed this population. Only one other tree, a different species, had mistletoe on it. And even though most of these trees were in that previous list of 50 that I had there. So what's going on, right? This mistletoe could grow on them in other parts of the U.S., but right there, it's not growing on those other trees. Interesting, huh? Let's take a look at this now. This is what you guys have in your hand being passed around the room. That's a shoot of mistletoe. Here's its fruits. And if you squeeze one of those, you probably got that viscid seed coming out on your fingers. That's the vicin that sticks it to the host branch. It then germinates based on light cues and other things, and it grows a little hypocotyl or radical out of the one end here, and that eventually orients towards and grows right into the host branch. It connects by means of what's called a haustorium. And after a year or two, it grows a little baby mistletoe shoot out of the branch at that point where it first entered. Now the question is, how's it recognizing things at this stage right here? And up until about 2 years ago, we had absolutely no idea how it's determining it's on a red maple or a silver maple or an American elm or whatever. But Chris Randall's lab did some fascinating work based on other work with dodder parasitic plants that they found were sensing their hosts by means of volatile chemicals. Right? These are monoterpenes, a couple of them shown here. So what he did was, very clever, you put the little seed in a dish and you put a little piece of filter paper with the odor in one direction, and you see whether or not the little radical or seedling root orients towards that smell. And you can see the one-way ANOVA test here with some of these, like limonene, myrcene, and philandrine. They're significantly different than control. But one of these monoterpenes, pinene, showed no difference from the control. So it doesn't like pinene, things you'd get from a pine tree. And by the way, this mistletoe never occurs on pine. But it is going after these others. So this is basically what he was doing, is measuring the orientation of that little growing radical deviation from the original position. So highly significant result. So this is the first clue that we have that mistletoes are smelling out their hosts. They're sniffing them. So in summary, this phorodendron is really a genetically diverse complex of populations. Many of these have been named at the species level. Morphologically, you can't really tell them apart very easily. They're hard to differentiate. The microsats gave us some level of differentiation, mostly between the East Coast, West Coast, but there's a lot of overlap, particularly in the state of Texas, where we get those all coming together. Now, when considered as a species, Phorodendron leucarpum has a very broad host range, but considered at the population level, only particular host trees are preferred. And we now know that these seedlings are orienting towards and actually attaching to the host based upon volatile host chemicals, monoterpenes. So I hypothesize that each mistletoe population then has a complex and unique array of alleles that are associated with host recognition and adaptation. So this complex really represents an active phase of speciation. It's really evolution right in front of our eyes. So that's my example of evolution in your backyard, our kissing mistletoe. Okay?